Dear students, welcome back to embryology classes, general embryology classes. So continuing with the first week of development. Where in previous session we saw fertilization. So today we are going to continue with the cleavage and blastocyst formation. So what is this cleavage? Cleavage is a repeated mitotic division of zygote. So we know at the end of fertilization we got this zygote as a result of fertilization, and zygote will start dividing immediately. So once it is formed, before it completely forms as a zygote itself, it enters into cell division. So it divides to form a 2-cell stage, 3-cell stage, 4-cell stage and 16-cell stage, all these cells. So these cells, they are surrounded by the glycoprotein layer, which is called as zona pellucida. The cells are compactly arranged within themselves. So the embryonic cells here, they become smaller with each successive cleavage division. So due to the division of the cytoplasm of zygote. So if you see here in this picture, so the size of the zygote is this big, and as we divide, the cells are arranged within the size of the zygote itself. Means only the cytoplasm starts splitting itself, and it forms into number of cells, smaller cells within the compact space. So the division of zygote begins 30 hours after fertilization. So after fertilization, it begins 30 hours after fertilization. After fertilization, where we can see the single-celled structure divides to form several celled stage, and the cells which are formed within the end product, these are pluripotent stem cells which are called as blastomeres. They are blastomeres. Blastomeres are pluripotent stem cells, means they have the capacity to divide and form any structure in the body. So such cells are called as stem cells. Pluripotent means highly dividing, rapidly dividing cells are called as pluripotent stem cells. So during cleavage, the blastomeres are within the zona pellucida. After 9 cells, so when the blastomeres here they divide, and when they form a 9-cell stage, the blastomere change their shape and tightly align themselves against each other, forming a compact cell mass containing around 12 to 32. Here we have written 16 cell, but it is not so specific to say as 16 cell. Usually we tell around 12 to 32 cell stage. So here it appears to be like a mulberry fruit which is called as morula. So here we can see the 16-celled stage is mentioned as morula, which is compactly arranged. So if we see the cavity of the uterine cavity, so this is, I am drawing the isthmus. Isthmus continues into the uterine cavity, and at the ampulla is the site of fertilization. So here is the zygote is formed here. So the zygote start dividing into a 2-celled stage. Slowly the 2-celled stage will divide to 3-cell stage, 4-cell stage. So they start dividing within it, and finally when they reach the 16-celled stage, it reaches almost near the entrance into the uterine cavity, and a 32-celled morula, that is the late stage morula it is called, it enters inside the uterine cavity. So almost around 4th day, so around 4th day, 3rd to 4th day we say, around 4th day the morula enters into the uterine cavity. So after this stage, the outer layer of the morula, is called as zona pellucida, which is a glycoprotein layer shell surrounds this compact cell mass, making the cell mass compactly arranged without gap. So in the uterine cavity we have got late stage morula. This is morula which reaches the uterine cavity. So now the morula has to further differentiates to form blastocyst, which also happens during first week of development. So what is morula? The cleavage of the zygote results a 16-celled stage. Not to be very specific, we can say around 12 to 32 cell stage called as morula. The morula enters the uterine cavity on 3rd or 4th day after fertilization. So that we saw just now. Now what happens? The zona pellucida, which is an outer layer, it starts disintegrating and it disappears completely. So disappearing of zona pellucida is called as zonal hatching. So what happens after the disappearance of the zona pellucida? A fluid, that is the fluid which is present within the uterine cavity, because the morula is now in the uterine cavity. So the fluid of the uterine cavity start entering inside in between the cells. Okay. So here in 32 cell, this is for example, so here we can say it is approximately 16 to 32 cell stage. So the outer layer zona pellucida is there. Here we can see the center cells are called as inner cell mass. Inner cell mass. The peripheral cells, the outer cells, called as outer cell mass. So now the fluid start entering into this, in between these cells, pushing the inner cell mass to one end. So making a fluid filled cavity which is like a cyst. So this cyst is called as blastocyst. So it is the cavity of the blastocyst. Now we can see here in the other picture, the inner cell mass is to one end. So this end is called as animal pole or embryonic pole. Whereas the opposite pole, where the cells are not there, here the peripheral cells are present. So the opposite pole is called embryonic pole or vegetative pole. And now because of formation of this cavity, the inner cell mass cells, they are now no more called as inner cell mass and outer cell mass. So the inner cell mass, now these cells are called as embryoblast. So this stage of conceptus is embryoblast. The outer cell mass is called as trophoblast. So the outer cell mass further differentiate into cyto and syncytio trophoblast. So we can now simply say it as trophoblast. And the initial 10 days of development known as blastogenesis, which forms the conceptus, that is blastocyst is formed. The conceptus will further differentiates to form embryo. So the beginning first stage up to 10 days of development, we see the development of conceptus. And the embryoblast cells projects into the blastocyst cavity, and the periphery, the blastocyst is formed by the trophoblast cells. So the outer cell mass will form the trophoblast cells, which forms the wall of the blastocyst. The zona pellucida gradually degenerates and disappears within the uterine cavity, and this is called as zonal hatching, and the blastocyst gains its nourishment from the surrounding uterine fluid. And as the fluid increases in the blastocyst cavity, it separates the blastomeres into two parts. A group of cells present centrally, or the embryoblast cells, inner cell mass, which eventually develops into embryo. So they are called as embryoblast cells. The outer cell layer is the trophoblast layer. In Greek, tropha means nutrition. So these cells will give rise to placenta. So trophoblast will later give rise to placenta, which provides nutrition to the developing embryo. So trophy in Greek, it means nutrition. So early pregnancy factors are released in this initial during 10 days of development. So an immune suppressant glycoprotein which is produced by the trophoblast cells, it appears in the maternal serum. So within 24 to 48 hours, there will be appearance of immune suppressant glycoprotein. And when we do blood test, we can check for the immunosuppressant glycoprotein to check pregnancy. And early pregnancy factor forms the basis of the pregnancy test, which is called a Rosette inhibition test. So during first 10 days of development, we can check whether the woman is pregnant or not with this early pregnancy factor. So that is immunosuppressant glycoprotein which appears in maternal serum. And second stage, further this blastocyst, now it has to undergo implantation. So we are going to see with the implantation. So the blastocyst attaches to the endometrial epithelium. Usually the site of embryonic pole. So if we see the blastocyst, we have got one side, the cells, which is the embryonic pole. This is the embryonic pole. And the peripheral cells are the trophoblast cells, or the outer cell mass will form the trophoblast. So this is embryonic pole. So usually the implantation occurs through the embryonic pole. So the blastocyst attaches to the endometrial epithelium adjacent to the embryonic pole, and which is the beginning of implantation. Normal site of implantation is posterior wall fundus of the uterus. To be more specific, posterior wall fundus, junction between the fundus and the body of the uterus. But normally we say it as posterior wall fundus of the uterus. So this is the anterior side of uterine cavity, where we are seeing the sagittal section here showing the slit-like uterine cavity. So this is the anterior side and this is the posterior side. And if you see the uterus, it has got the fundus, the upper projected part, fundus, which is present above to the level of uterine tube, and the central part is the body, and body continues to form cervix. Cervix. So we can see here posterior wall at the junction of the fundus and the body. So this is the site of implantation. So implantation of the blastocyst, it begins during the at the end of the first week, that is around 5th or 6th day of development, and the implantation completes by 10th to 11th day after development. So we can just write as 6 to 10 days. So it begins on 5th or 6th day and completes by 10th or 11th day. So implantation begins in the first week and continues into the second week of development. So it is during 6 to 10 days after ovulation. So after ovulation you can calculate the implantation occurs during 6 to 10 days. So as soon as the implantation begins, the trophoblast starts differentiating into outer syncytial trophoblast and inner that is the cytotrophoblast. So the type of implantation in humans, it is interstitial implantation, means the complete blastocyst it enters into the endometrium fully, and it gets embedded within the wall of the uterus fully. So this type of implantation is interstitial implantation, which is happening in humans. Whereas if you see in other animals, part of the blastocyst is eroding the endometrium, means part of the embryo will be inside the endometrium and part of the embryo is developed outside within the uterine cavity. So partially it is embedded within the endometrium. All through the pregnancy it is like that. In humans it is completely embedded within the wall of the uterus. That is called as interstitial implantation. So this is how the embryo conceptus completely gets embedded within the wall of the uterus, that is in the gets embedded with where we can see after embedding the site where it gets embedded, it gets closed, forming a complete scar, filling up with the fibrous tissue. And within first week you can see one more change, that is the trophoblast cells. The trophoblast cells, they get differentiated into inner cytotrophoblast. So here the cellular layer which is present here, these cells are present individually. This layer is called as cytotrophoblast. Whereas the outer part of the layer, the cells start fusing with each other. So we are seeing there is no cell membranes here, only the nuclei is present like a jelly-like substance, which is called as syncytiotrophoblast. Syncytiotrophoblast. Cytotrophoblast is the inner layer and which contain actively dividing cells and which rapidly divides by mitotic division, and newly formed cells migrate into increasing mass of syncytiotrophoblast, where the cell membranes are lost here and the cells fuse with the multinucleated appearance. So we are seeing multinucleated, this gel-like substance which is yellow color represented. So that is syncytiotrophoblast. Syncytiotrophoblast contains a multinucleated protoplasmic mass with no cell membranes. And syncytiotrophoblast extends through the endometrial epithelium and invade the connective tissue. So here this is endometrium. So it invades the connective tissue of the endometrium and through the endometrial epithelium and invades the connective tissue. And further the syncytiotrophoblast expands surrounding the embryonic pole. So this is the embryonic pole where the embryonic cells are arranged in two layers now, whereas the we can see the syncytiotrophoblast is more prominent on the embryonic pole than on the abembryonic pole. So the summary of the first week of development, we saw the gametogenesis, means formation of the spermatozoa and formation of the female oocyte, that is matured oocyte, which is arrested in the secondary oocyte stage by oogenesis. So both spermatogenesis, oogenesis is considered under gametogenesis. And during first week of development, we have completed about fertilization, and we saw the cleavage where the zygote divides to form a mulberry fruit-like structure called as morula. So we saw the development of morula, and now the morula turns to form a blastocyst having a cavity inside it called as blastocyst cavity, which is a fluid-filled cavity having inner cell mass and outer cell mass, and inner cell mass will form embryoblast and outer cell mass will further differentiated into trophoblast. And trophoblast also we saw it is differentiated into two layers, that is the inner cytotrophoblast and outer syncytiotrophoblast. And this syncytiotrophoblast and cytotrophoblast, they invade the endometrium where the complete blastocyst is implanted. So during the first week, the beginning of implantation occurs and completion of implantation is by 10th or 11th day. That is during second week of development, the implantation is completed. So suppose if this blastocyst, instead of getting implanted in the normal site, that is fundus, posterior wall fundus of the uterus, suppose if it implants anywhere else, so it results in different positions of placenta and ectopic pregnancies. So it can be tubal pregnancy. Suppose if it is implanted within the tube, that is the uterine tube, and it can be ectopic completely out of the uterus, that is within the peritoneum. It can happen, and sometimes the implantation may occur inside the ovary, that is called ovarian implantation. and sometimes we find it gets implanted within the uterus but not on the fundus of the uterus, the other parts of the uterus, which results in a condition called placenta previa. So now we know the normal site of implantation is the posterior wall fundus of the uterus. Instead of here, suppose implantation occurs in the lower part, that is in the body of the uterus. So now this placenta previa we can consider in 3 phases based on its severity. So if it is somewhere in the body of the uterus, that is in the uterine cavity, within the uterine cavity it gets embedded within the wall of the uterus, but in the body, not in the fundic level. So that is called as first grade. And if the implantation occurs in the lower segment and partially covering the internal os, that is the second grade. And most severe grade is the third grade, where the implantation occurs covering the internal os. That is the third grade of placenta previa. So in third grade, what happens during delivery, during normal, suppose if the woman is not diagnosed previously about this placenta previa, and if she undergoes normal delivery, the placenta, as it is present in the internal os, the first structure which ruptures is the placenta, and we know the placenta is a highly vascular structure, so there will be a severe loss of blood, and the mother may die because of hypovolumic shock. And sometimes if it is in the partial covering the internal os and all, there also there may be a chance of rupturing of the placenta. And this position is not sometimes, even though it is implanted in the lower segment, as the pregnancy continues, it may ascend up, the placenta may ascend up. But if it doesn't ascend, it may result in placenta previa. So this is a small idea about placenta previa. So this completes the first week of development. Thank you.